Cellular respiration is the complete breakdown of a glucose molecule in order to produce the energy molecule adenosine triphosphate, or ATP. This reaction can be written as the addition of glucose and oxygen to form carbon dioxide and water. Cellular respiration is a catabolic reaction, or a reaction in which large molecules are broken down into smaller ones. In cellular respiration, there are three major carbohydrate energy-producing reactions: glycolysis, the citric acid cycle, and the electron transport chain. All three reactions work in sequential order to produce a net yield of 36 molecules of ATP. However, in glycolysis alone, the goal is to just produce a net yield of 2 ATP. Glycolysis is an anaerobic process, meaning it doesn't require the use of oxygen. Glyco means sugar and lysis refers to breaking, so glycolysis is literally the breaking down of sugar. This breakdown forms what is known as pyruvic acid. Glucose is a necessary sugar for energy production so that our bodies can function soundly. There is a supply of glucose that can be utilized by the brain, but the supply is very limited. Therefore, there is always an amount of glucose stored in our blood. Specifically, glucose is a carbohydrate molecule and consists of 6 carbon atoms connected as a chain. Overall, cellular respiration occurs in the mitochondria, but glycolysis specifically occurs in the glue-like cytoplasm of cells. Once a 6-carbon atom glucose molecule enters the cytoplasm of a cell, a phosphate group attaches itself to the glucose via a hexokinase enzyme. This enzyme accomplishes this by taking phosphate away from ATP, converting it to ADP, which is adenosine diphosphate. The hexokinase enzyme delivers the phosphate to the glucose molecule, and the phosphate attaches itself to the end of the molecule, making it a glucose 6-phosphate. This process is called substrate-level phosphorylation. This is important because it will keep the glucose from leaving the cell, since attached phosphates cannot permeate the cell membrane. The enzyme phosphoglucoisomerase forces the glucose 6-phosphate molecule to rearrange itself to form fructose 6-phosphate. Then the enzyme phosphofructokinase causes another phosphate group to attach to the molecule. This uses up a second ATP molecule, and it ends up forming fructose 1,6-biphosphate. Next, the enzyme aldolase makes the fructose 1,6-biphosphate split into 2 molecules. Since the original glucose molecule had 6 carbon atoms, the 2 new molecules are each now 3-carbon chains, known as DHAP and PGAL, more commonly known as G3P. The enzyme isomerase converts the DHAP molecule into a G3P itself, and there are now 2 G3Ps. The G3Ps each gain phosphate groups while simultaneously contributing 2 electrons and a hydrogen ion to the low-energy coenzymes NAD+. NAD+ accepts and donates electrons. NAD+ will form 2 NADH, which are high-energy carrier molecules. Also, the added phosphates are used to regain the ATP that was lost in the first stage of glycolysis. Therefore, the molecules each have just 1 phosphate again, centered at the middle of the 3-carbon chain. These new compounds are called pyruvates. They will eventually be absorbed by the mitochondria, and a carbon dioxide molecule is removed from each pyruvate as the rest goes on to the citric acid cycle. The enzyme phosphoglyceromutase changes the phosphates group positions, and the molecules are now known as phosphoglycerates. Enzyme enolase then forces the phosphoglycerates to each lose 1 water molecule, and this leads to phosphophenol pyruvate. Recall that each of the 2 3-carbon chains have a centered phosphate attached to them. The centered phosphate of each of the 2 chains is finally transferred to ADP molecules to form 2 whole ATP molecules. It is here the process of glycolysis finally reaps its rewards by producing a net gain of 2 ATP. Pyruvic acid is the end product of glycolysis, and after glycolysis, acetyl CoA, or acetyl coenzyme A, is a molecule formed, and it carries the carbon atoms of the acetyl group and initiates the citric acid cycle. which is the next reaction of cellular respiration following glycolysis. Thank you for watching.